be showing you how to grow and immobilize bacteria, and the first step in this process is making an agar plate with the bacteria streaked on it. And so we keep all of our bacteria in stock samples in the negative 80-degree freezer. So I just need to grab that out of the freezer and bring it over to the bio hood. Okay, this is what the freezer stocks look like. And just a reminder, before working in the bio hood, we need to clean our hands, and so we just squirt some 70% ethanol to make sure everything's sterilized. Alright, so I have my agar plate here. And what I'll do is I'll take these sterile sticks that we have, and I'll just take one swipe from the inside of this stalk. There's a little bit on the tip there, that's all you need. And I'll go ahead and streak that there. And that's it. So now the agar plate is streaked, and we'll go ahead and wrap this up with parafilm and put it in some aluminum foil and just let it sit. The bacteria should be grown in about a day or so. Then this stock just goes right back in the freezer. Okay, the E. coli has grown on the agar plate, and now we can harvest a single scoop or a single culture from this plate and put it into our solution to grow in an overnight culture. So to do that, first we need to take 150 milliliters of this LB broth, and this is just a broth, a type of solution that these E. coli cells can grow really well in. And so here we have a sterile growth flask here, and we're just going to take our sterile pipettes and our automatic pipetter and measure out 150 milliliters. Yeah! Okay, so we have 150 milliliters of LB broth in here, and now I'm just going to use one of our sterile blue sticks and take a scoop from the agar plate and dissolve it in this solution. And when you're choosing where to scoop here, you just want to get one single colony, and so I'm just going to scoop a little section right here. That's all you need. And then swirl it around and swirl the flask itself to make sure everything is off of the stick. And that's it. This is going to go on the shaker, and it's going to incubate for about 24 hours, and we'll check on it in the morning. So here is the overnight growth culture here, and I'm just going to take it out of the shaker and bring it over to the bio hood and start our immobilization process. And here the E. coli culture should be cloudy, but not have any visible white floaties or other contaminations. Okay, so we have our overnight culture here, and I'm going to take 50 milliliters from here and put it in each falcon tube, which we put into the side of the centrifuge. And to do this, we use these sterile pipettes here and the automatic pipetter. And here I just have 50 milliliter sterile pipette. And when we're done with the pipette tip, we put it back in the holder, and we use a fresh one for every culture. Okay, so this can go back on the shaker, and then these will be put in the centrifuge. So here we have our centrifuge baskets, and I've just put each of our E. coli samples in each one. Now in the centrifuge, they need to be exactly counterbalanced as to avoid any explosions in the centrifuge. And so how we do that is we have two falcon tubes here that we put in, and these just have water in them. They just say water here. And what we'll do is we'll balance it on the mass balance and make sure that they're within 0.01 grams. So I'll show you how we do that. First, we put one on the balance, and we're just going to go ahead and tare that. And so this reads 0.00 grams. And then we will adjust this one, we'll adjust the water so that this reads as close to 0.00 as possible. Okay, so now we are within 0.01 grams, which is how close you actually do have to be. Awesome. So they're exactly the same weight, and so now we can put them in the centrifuge. Make sure everything's capped as tightly as possible, and then you set these baskets in opposing sides so everything is counterbalanced there. Push down on the lid, and here are samples. Like to run at 4,500 RPMs for 20 minutes, and mine is at 4 degrees Celsius. And to start this, we just click, click. While the bacteria is centrifuging, I can get some of the rest of my materials ready, including the little 1.5 ml tubes that I'm going to use for the next step. And so here we're just going to take 2 Eppendorf tubes at 1.5 mils, and I'm going to pre-weigh them because afterwards we're going to put our cells inside these tubes and then get the end mass for that. And so in order to get the correct mass, we need to pre-weigh these on the mass balance. So here I am taking note of the weight of the tube in milligrams and writing it directly on that Eppendorf tube in order to keep track. Another note here is that we always round to the nearest milligram. Also, while the centrifuge is running, I can collect the other items that I will need for this bacteria immobilization process. In the biosafety hood, I've put my 2 pre-weighed Eppendorf tubes, the buffer solution that I'll be using for resuspension, a 1000 microliter adjustable micropipette with sterile pipette tips, and the sterile blue sticks. I have also put 2 beakers, one for liquid waste, and one to put used pipette tips and sticks in.
Here is what the cells look like after spinning in the centrifuge. As you can see, they are all concentrated at the bottom and the LB broth sits on top. This is called the supernatant. And this LB can be quite noisy and interfering in electrochemical experiments, so we get rid of it by dumping that supernatant in a waste beaker and doing a washing step. In this wash step, we resuspend the cells in 1 milliliter of buffer, and that goes directly into the falcon tubes containing the concentrated cells at the bottom, and we centrifuge again. In order to fully wash and incorporate the buffer with the cells, vortex the tubes until the solution is completely mixed and there are no chunks of cells left. In this second centrifuge step, we will be transferring the cell and buffer mixture to the small 1.5 milliliter Eppendorf tubes. To do this, transfer the solution from one falcon tube to one Eppendorf tube using the micropipette. Repeat the same process for the second set of tubes. The Eppendorf tubes with the cell and buffer mixture can then be carried to the small centrifuge for a second spin step. Since the volumes and the weights are so small, they don't have to be exactly the same weight, but make sure to counterbalance them in the centrifuge by placing them in opposite spots. This centrifuge step will run for 10 minutes at 4 degrees Celsius and 15,000 RPMs. After the cells are done spinning, they are once again concentrated at the bottom of the tube with the buffer solution lying on top. Now the cells are effectively rinsed and free of their LB growth medium. The next step here is to dump that supernatant in a waste beaker so only the concentrated cells remain in the vial. For the immobilization procedure, we want 1 microliter of buffer per milligram of cell, which gives a cell density of 1 gram per milliliter. To do this, we weigh the cells plus the tube and subtract the mass of the pre-weighed tube to find just the mass of the cells. Back in the biohood, you can adjust the micropipette to the number of microliters that the cells weighed in milligrams. For example, here my cells weighed 209 milligrams, so I will suspend them in 209 microliters. So now the cells are at the desired density of 1 gram per milliliter, and all we have to do is thoroughly mix the cells and the buffer. But because the cells are so sticky, it helps to manually stir the solution with one of the blue sticks before taking the tubes to the vortexer. With the mixed cell solutions, it's finally time to immobilize the bacteria on an electrode. Here I am immobilizing 60 microliters of cell solution onto carbon paper electrodes. I use a micropipette that is set to release 60 microliters and carefully place the bacteria droplet in the middle of the electrode. Since I am working in an aqueous buffer solution and the surface of the carbon paper is hydrophobic, the cell solution will form a droplet as opposed to completely spreading out over the electrode surface. Final step is sealing the desiccator with a vacuum line and running a slow stream of nitrogen to dry the electrodes before using them in any experiments. Drying time will vary depending on the amount of cells deposited on the electrode, but these typically take an hour to dry.
Did not get your thumbs up at all.